So when thinking about physiology, we're going to move through the whole alimentary canal, one segment at a time. And for each part, we need to think about what's being secreted, what digestion is happening, what absorption is happening, and what motility is happening. So we start in the oral cavity, and in the oral cavity, we are secreting saliva. So the secretion is saliva. Saliva contains many things that are going to be important. It's got mucus, which helps with lubrication of the bolus that you're forming. It's got salivary amylase. Amylases digest carbohydrates, so this will actually begin carbohydrate digestion in the mouth. Saliva also contains stuff called lingual lipase. This is not activated until it gets to the stomach, so this is not active. We won't begin lipid digestion in the mouth. Some other things that are present, there's this stuff called lysozyme, which is an antimicrobial enzyme, and so that can help just with providing some innate immunity. And there are also some antibodies, but we didn't go over the specific different types of antibodies, so just be aware that there are antibodies also present in saliva, and that's going to help with the innate immune response in the mouth, so that if you ingest a pathogen or foreign particle, we can either deal with it with lysozyme or with antibodies. So that's what the oral cavity is secreting. As far as digestion goes, again, we'll begin. Well, we're going to start our mechanical digestion with our grinding and tearing of food. So we can say there's some mechanical digestion with grinding and tearing. As far as chemical digestion goes, the only real digestion that's going to happen is with our carbohydrates because of salivary amylase. So we'll say we begin carbohydrate digestion in the mouth as well. As far as absorption goes, your entire GI tract can absorb water, and all of your cells can use glucose. So if you have very simple sugars, just glucose, then you can absorb that as well. But there's not going to be significant nutrient absorption of any kind. So for absorption, we'll say some water. You can absorb some glucose, and that's just because every cell can use glucose. If it's a complex carbohydrate, we're not going to break it all the way down to glucose in the mouth. But like I'm drinking this fruity drink here, I'm sure that it's got not just fructose but glucose in it. I could absorb that in my mouth. But as far as other nutrients, we'll say no significant nutrient absorption. As far as motility in the mouth goes, we've got the first phase of swallowing. Swallowing is also called degglutination. So, degglutination equals swallowing. And there are 3 phases to swallowing. Your book breaks swallowing down into 3 phases. The first phase is the voluntary phase, and that occurs all in the oral cavity. So what happens during this first phase, our voluntary phase, is that you've mixed your food with saliva. You've formed a nice bolus out of it. You've chewed it up so you feel it's chewed enough, and you'll start pushing it along your hard palate. So, and really getting that bolus into a nice swallowable piece. And as you push it to the back of your hard palate, it's going to hit your soft palate and stimulate tactile receptors. That's the whole voluntary part of swallowing, is forming that bolus and pushing it back to your soft palate. As soon as we hit those tactile receptors, then we're going to push the food bolus back into our pharynx, and that'll begin the second stage, which is involuntary. So for our first stage, motility in the mouth, we can say that's the voluntary phase of swallowing. And what's happening is your tongue is pushing the bolus along your hard palate back towards the posterior aspect of your mouth or your oral pharynx. So you're going to start pushing it back to get ready to swallow it. And we'll say until it hits soft palate. Once it hits that soft palate, and we stimulate those tactile receptors, we're going to enter the second phase of swallowing, the pharyngeal phase. And the pharyngeal phase is involuntary. So what happens during the pharyngeal phase of swallowing is your soft palate and your uvula are going to raise up and protect your nasopharynx, so food doesn't go up your nasopharynx. Your epiglottis is going to flop back and cover your larynx, so food doesn't go down your larynx. And then the food will pass into your pharynx, through your pharynx to that upper esophageal sphincter. So we'll say uvula and soft palate lift. And this blocks the nasopharynx. We'll say the epiglottis covers your larynx. And your food enters your pharynx. And it's going to go to that upper esophageal sphincter. So that's our pharyngeal phase. The uvula raises and soft palate raised to block the nasopharynx. Epiglottis covers your larynx. Food goes down your pharynx until we hit that upper esophageal sphincter. The third phase of swallowing is also involuntary. This is called our esophageal phase. And our food is going to pass through that upper esophageal sphincter, and it's going to be moved by a peristaltic wave down ultimately till we get to our lower esophageal sphincter. And what happens as that peristaltic wave is moving down the esophagus, it's going to cause that lower esophageal sphincter to open. So we'll say that our food passes, I should stop calling it food, it's a bolus. Our bolus passes the upper esophageal sphincter, and is moved by a wave of peristalsis. This wave of peristalsis is going to relax the lower esophageal sphincter, also known as the cardiac sphincter, so that food can pass into the stomach. So if we think about our pharynx and what it is absorbing, secreting, digesting, as far as secretion, our pharynx is secreting mucus. It's not really doing any more significant digestion. Carbohydrate digestion continues. Again, it can absorb water and glucose, but no significant nutrient absorption. And the motility here, we would say, would be our pharyngeal phase of swallowing. For our esophagus, pretty much the same thing. Our esophagus is secreting mucus. Carbohydrate digestion continues. We're absorbing some water and maybe glucose if those cells need it. And the motility is the esophageal phase of swallowing. And we could specifically say that if you wanted to describe it a little more, you could say that this peristaltic wave is what's happening for our motility, and it's going to relax that lower esophageal sphincter. All right, when we get to the stomach, our secretions in the stomach are coming from the cells in our gastric pits. And our gastric pits are just those regions where our columnar epithelia kind of dips down into lamina propria. And all at the top of our gastric pits, we have those mucus neck cells. And these are secreting mucus that's helping to establish the mucosal barrier in the stomach that protects it from digesting itself. On the surface here also there are just what are called mucus cells. They are also secreting mucus. So the stomach has two types of mucus secreting cells. Mucus cells on kind of facing the lumen, and mucus neck cells at the superior aspect of your gastric pits. Other cells that we find in our gastric pit are going to be chief cells and parietal cells. So we'll have parietal cells. These are secreting hydrochloric acid. And we'll have chief cells. These are secreting our pepsinogen. I should also say our parietal cells are secreting this stuff called intrinsic factor. Intrinsic factor helps with the absorption of vitamin D in the small intestine. At the very bottom of the gastric pit, you'll find these things called G cells or enteroendocrine cells that are releasing gastrin, but we're not really going to talk about gastrin. We're not going to go over the regulation of digestion a whole bunch because you guys have enough to worry about. So, and that's part of what's going on there. We're going to talk a little bit about one hormone coming from the small intestine because it's important for getting other digestive secretions to the area. So as far as secretion goes, we could say that the stomach is secreting gastric juice. So the stomach secretes gastric juice, which is a mix of all this stuff except for our gastrin. It's a mix of our hydrochloric acid, intrinsic factor, pepsinogen, and actually our chief cells are also releasing something called gastric lipase. So for secretion, this is gastric juice. That's made up of all of this stuff, our hydrochloric acid, our intrinsic factor, our pepsinogen, and our gastric lipase. All of that's found in gastric juice. So for digestion, we can say that there's actually going to be some mechanical digestion as we're mixing and churning and mixing that bolus that came into the stomach with this gastric juice. That counts as mechanical digestion. So we have mechanical due to mixing and churning. This is also motility for the stomach. It's got these powerful contractions that are going to mix that bolus of food, and it's because it's got that extra layer of muscle, the oblique layer, so that we can have these nice forceful contractions that mixes that bolus with gastric juice. So mixing and churning is going to help with mechanical digestion. As far as chemical digestion goes, our hydrochloric acid is going to activate lingual lipase. So we can take big fat droplets and get it down to littler fat droplets. We're still not really going to be breaking apart the fat droplets. We need bile salts for that because fat droplets are hydrophobic, and this is a hydrophilic area. But we can kind of begin lipid digestion. With our lingual lipase that becomes activated. So we'll say with active lingual lipase and gastric lipase. So hydrochloric acid is what's activating our lingual lipase. Hydrochloric acid is also going to denature proteins. So that'll help get these proteins ready for digestion. So as far as the things that hydrochloric acid is doing, it's going to activate lingual lipase. It's going to denature proteins. So that'll take them from these complex quaternary structures down to their tertiary or secondary structures that we can then, the third thing that it's going to do is it's going to activate pepsinogen to become pepsin. So we'll say that it's going to convert it to pepsin. And now pepsin can do protein digestion. So we'll start breaking those denatured proteins down into polypeptides. So that's another thing that will happen here, is we'll say we're going to denature proteins and pepsin is going to break them into polypeptides. All right, so for motility in the stomach, we've got mixing and churning. And again, these are forceful contractions say, aided by the oblique layer of the muscularis externa. And these are going to mix the bolus with gastric juice. Next we have peristalsis. This is like peristalsis anywhere. This is just the contractions that propel the material toward the small intestine. This material, once we've really mixed that bolus of food with gastric juice, this material becomes called chyme. So peristalsis will move the chyme towards our duodenum of our small intestine. Once that food's nice and mixed, it's going to start, we're going to have these waves of peristalsis that are going to push it towards the pyloric sphincter. The pyloric sphincter opens about every 15 minutes and allows a little bit of chyme through, and then it snaps shut. When it snaps shut, it causes another kind of motion in the stomach that we call retropulsion. And so that is when our food will kind of blow back because of the closing shut of the pyloric sphincter, so it moves back up toward the body of the stomach. So retropulsion is like our, after the pyloric sphincter snaps shut, the food will move back toward the body of the stomach. Then 15 minutes later, we'll get more peristalsis that pushes it toward the pyloric region. Pyloric sphincter opens, put a little chyme through, snap shut, more retropulsion. The mucosal barrier that's formed in the stomach is actually, we've got the mucus doing it, and then also our cells are joined by tight junctions, so nothing really goes through. So there's not going to be significant nutrient absorption in the stomach. Again, if the cells need glucose, they could take that in, some water absorption, but no significant nutrient absorption. All right, then from the stomach, we will move to the small intestine. So the small intestine is going to be our major site for digestion and absorption. And the small intestine is actually going to be secreting a couple things. It secretes this hormone called cholecystokinin. So for our small intestine, and this is the only hormone I really want you to be aware of. It's cholecystokinin, or CCK. And what CCK does is it's going to stimulate gallbladder contraction. And when the gallbladder contracts, it's going to squirt bile into the duodenum. So CCK stimulates gallbladder contraction, and the gallbladder then will squirt bile into duodenum. CCK is also going to stimulate the pancreas to secrete pancreatic juice into the duodenum. So CCK is stimulating these other two organs to add their secretions to the small intestine. The small intestine is also secreting what are called brush border enzymes. And our brush border enzymes are a mix of proteases, nucleases, and amylases that are going to be able to finish up chemical digestion. So they do the last part of chemical digestion. Bile is going to help with lipid digestion because bile salts form micelles around lipids. Micelles are, it's like how detergent works when you stick it in your oily pan. Micelles wrap around our lipid-soluble or water-insoluble materials and create a hydrophilic shell that will make them be able to mix with water. So bile salts are going to wrap around lipid droplets, and now our lipases can get in there and start breaking the fats down. So our bile salts form micelles around lipid droplets, which is going to be important for lipid digestion. Our pancreatic juice contains a bunch of different things. It contains pancreatic amylase, which is going to be important for carbohydrate digestion. Pancreatic amylase is going to take polysaccharides, and it's going to get them down to like disaccharides. So our salivary amylase got really complex starches down to this polysaccharide molecule in the stomach. So if you've got a really complex starch, salivary amylase starts the work. And then we'll have these polysaccharides that enter the small intestine, and pancreatic amylase can break them down into disaccharides. So for carbohydrate digestion in the small intestine, it's going to begin with pancreatic amylase. Our brush border enzymes are a mix of everything else. So they've got amylase, they've got proteases, which break down proteins, they've got what are called nucleases that break down nucleic acids. And so now, for our carbohydrate digestion, our brush border enzymes are going to take these disaccharides down to monosaccharides. So our brush border enzymes are going to digest our disaccharides to monosaccharides. And then we will absorb those monosaccharides. So we'll say this is absorption. Monosaccharides are absorbed into the blood. That's going to go to the hepatic portal system. So for carbohydrate digestion, you'll take in complex carbohydrates in your mouth. Salivary amylase will start breaking them down to polysaccharides in your stomach on its whole journey down. Then when the chyme enters your small intestine, it'll be polysaccharides that you have. Pancreatic amylase breaks those polysaccharides into disaccharides, and then the brush border enzymes finish up carbohydrate digestion by breaking our disaccharides into monosaccharides. Monosaccharides are what we can absorb, and they'll be absorbed into the capillaries that are going to go off and be filtered through the hepatic portal system. All right, for lipids, again, proteins. Let's do proteins first. So pancreatic juice is going to contains a bunch of different what are called proteolytic enzymes. And I think your book goes over the specifics. I'm not going to test you on the specifics, but just be aware proteolytic enzymes are going to break down proteins. So in the stomach, we had these complex proteins that got denatured, and then we broke those down into polypeptides. So now these proteolytic enzymes are going to be able to take our polypeptides and digest them down to peptides. Our brush border enzymes are going to take those peptides and break them down to amino acids. And then that's what we can absorb, the amino acids. So, and our amino acids, again, are absorbed into the blood. So you eat a piece of steak in your mouth, you start chewing it, you get it to your stomach, the hydrochloric acid denatures those complex proteins. Then activated pepsin will start breaking those proteins into polypeptides. Polypeptides are part of chyme then, enters your duodenum. Pancreatic peptidases or proteolytic enzymes are going to break those polypeptides down into peptides. And then our brush border enzymes are going to finish protein digestion by breaking the peptides into amino acids. Amino acids are what we can absorb. They're absorbed right into the blood, capillaries in the small intestine, taken off to the hepatic portal system for filtering. All right, lipid digestion is more complex because lipids are not water soluble. So the first thing that we have to do is wrap them up in these micelles that are going to make them accessible to our lipases. So for our lipids, the first thing that's going to happen with our lipids is they're going to be entering the stomach as these really big fats. And so our gastric lipase and lingual lipase are going to be able to get them down to little fat droplets. So then when they're entering the duodenum, they are fat droplets. And the bile salts are going to surround these fat droplets and create a micelle. And now our pancreatic lipase can get in there and start breaking these fat droplets down to free fatty acids and glycerol. So our bile salts create a micelle, and then we've got pancreatic lipase can break the droplet into free fatty acids and glycerol. These micelles are going to be absorbed through the cells in the small intestine and become these things called chylomicrons. So this is where it's going to be a little different. We're not absorbing straight to the blood. They're going to our little they're still micelles, but now it's not these big complex fat droplets anymore. They're micelles with our free fatty acids and glycerol. And these are absorbed through our small intestine cells and become what are called chylomicrons. Chylomicrons are absorbed in lacteals. It's FFA and glycerol. So this first micelle is just a big fat droplet with salts around it. But as we let the pancreatic lipase in there, it's going to break that fat droplet down into free fatty acids and glycerol. It's still a micelle because it's got our bile salts around it, but now it's got free fatty acids and glycerol in it. That's going to be able to be absorbed through our small intestine cells, and then that structure becomes what we call a chylomicron. And this is what we're absorbing into the lacteals. So it gets absorbed through the cells in your small intestine and becomes these things called chylomicrons, and that's what we're absorbing in our lacteals. So our pancreatic juices are really helping to kind of be this intermediate step between our large macromolecules that were in the chyme and getting it down to where our brush border enzymes can take smaller molecules and convert it to our monomers. Monomers is what we're absorbing. So we definitely have to get our pancreatic juice and our bile into the duodenum, so CCK is going to help with that. Then our brush border enzymes are secreted by those by the brush border, and that is going to be like the final step of all of our digestion. We can also digest nucleic acids. As far as motility in the small intestine goes, we have peristalsis, which is going to propel the material toward the small intestine. So it propels material toward large intestine. Some books still call it chyme, but stuff's happening to it, so it's slightly different than chyme. I would just call it material. There's a second type of motility in the small intestine called segmentation. And segmentation are like these small kind of side-to-side contractions that are going to help to do several things. It's going to help to, we'll say these are small side-to-side contractions. That are going to first mix the material with our accessory gland secretions, and then also it's going to help to expose new parts of the material to the brush border. So as we're kind of shaking back and forth, then we can expose new parts of the material to the brush border. And that's going to serve two purposes. One, to mix with our brush border enzymes. And then also to help to expose to the absorptive surface. Those same cells are doing the absorbing of our nutrients. So we'll be able to increase absorption of nutrients. And then peristalsis is going to be moving it all forward. So as we reach the most distal part of the small intestine, the ileum, then this peristaltic wave is going to relax that ileocecal valve, and material can pass to the large intestine. All right. So for our large intestine, as far as digestion goes, it's not doing any digestion of its own. So what's it secreting? It secretes mucus, and it secretes a lot of mucus. So the material that's passing through here, we're going to absorb a lot of water from it. A lot of our water was absorbed in the small intestine, so we can't forget that's happening. Our small intestine's absorbing our monosaccharides, it's absorbing amino acids, it's absorbing these micelles with their free fatty acids, it's also absorbing water, it's absorbing vitamins and minerals. So most of our digestion and most of our absorption happens in the small intestine. So when we get to the large intestine, this material is mostly solid. So we really need to be able to lubricate it, so there's a lot of mucus. And when you looked at the histology of the large intestine, you probably noticed there are a ton of goblet cells in it, because it's very busy secreting mucus. As far as digestion goes, it doesn't do any digestion of its own. So for the large intestine, it's not doing anything. But we have bacterial flora, and they are digesting all of your undigested material, or indigestible material. And then they are going to release vitamins B and K. So for absorption, then in the large intestine, we're going to be absorbing a lot of water and vitamins B and K. For motility in the large intestine, we have peristalsis, which does what it always does. It's propelling the material forward, and it's propelling it towards the rectum. We also have two other types of motility in the large intestine. First, what are called haustral contractions. And you may remember that each of these little segments in the large intestine is called a haustra. So each of these little kind of nicely divided off segments is a haustra. And haustral contractions is a contraction within one haustra that moves material to the next haustra. And then we'll have another haustral contraction and it'll move the material to the next haustra. So haustral contractions move material from one haustra to the next. And then the last type of motility in the large intestine is what's called a mass movement. And these happen 1 to 3 times a day following a meal. So as you're eating or following a meal, you'll have a mass movement. And what this is a very strong, forceful contraction that originates about halfway through the transverse colon. So it's a really forceful contraction that's going to propel material through your colon and toward the rectum, so that you can make movement, make way for all the new material that you're bringing in. So we'll say this is a forceful contraction originating in the transverse colon that propels mass amounts of fluid toward the rectum.